Well, good afternoon. I'm here just to talk about also emerging contaminants and some microbial communities, not in the depth that Nora has done before. And I would like to join her crusade to convince people that, if necessary, that emerging contaminants exist, they are there, and they can reach interesting levels that makes us wonder about them and take care about it. That's a project, the European project, okay, that many people intervene in it. We are two of the authors here, but it's me who is going to do the presentation. Tough questions can be addressed to the postdoc researcher, that, as usual, she's the one who knows all the details. So how does this work? Is next page down. Okay. Our idea is not just to go to an aquifer, that I'm going to introduce later on. It's not just to take samples and describe them. But the problem we faced when we presented this project was to understand how those emerging contaminants, and in particular those antibiotics, those who enter the system through the use of swine manure fertilization, how they migrate in the subsurface, particularly in the saturated zone. That was the goal. It was not just to say, well, we have these pollutants here, but we want to know, we ask ourselves, what are the mechanisms? If they are hydrological, if they are geochemical, what does control this? I think that what we are going to present, okay, what we're going to present probably is very simple, but for us was quite a revelation to say, I think that what we read in, not in the scientific literature, because there is not much literature on that, but what we read in the official documents, they don't reflect the reality of what's going on inside the aquifer. So our idea, the project is called Persist. So because we thought it was very difficult, we say, wait, keep calm and do it, and let's see what happens. And the idea was to say this migration of the antibiotics, to discuss why their spatial distribution is so strange. And finally, if you look at antibiotics, you have to look at the resistance of these microbial communities that exist in groundwater, and that they can also pose a serious health concern. So I'm going to talk more or less about the two first, but I also want to introduce our results on the third topic. The aquifer is located in northern Catalonia. It's a small, there is no scale, but the red square will be probably 15 kilometers by 15 kilometers. That's 10 miles more or less. And it's a fluvio-deltaic aquifer. From a local river, its discharge is very low. It's a Mediterranean river at the end, and there is a fluvio-deltaic system, as I said, and we just worked on the upper part of the aquifer. We selected those wells which are related to the water table aquifer, and they don't go farther than 15, 20 meters depth at the most. It's quite an homogeneous gravel sand system, so we can say that we are not going to find geological heterogeneity. Okay, that's very simple to say, but we believe that's quite a homogeneous type of aquifer. In the field work, we took samples, 47 samples in wells, 7 samples in the river, which are going to be interesting. Then we took also 2 picker slurry samples. I don't advise you to go and take those samples. It's not the most rewarding work you can do. Geophysical surveys, and now we are doing some monitoring of 8 representative wells on antibiotics and antibiotic-resistant genes, which are not going to be shown here. Our colleagues in the lab analyzed every groundwater sample for a set of 53 different antibiotics, and the microbiologists did very complicated things with the samples, which I am not able to explain to you in detail. I put this image here just to show that we are in an agricultural area. An agricultural area that has a lot of swine livestock, so all the manure has to be used as a fertilizer, used or recycled or misused. I don't know exactly the name, okay? But it's applied. You have 2 types. You have orchards, mainly apples, which are irrigated, but you don't apply the manure to the orchards. And then we have wheat in winter, corn in summer, which means that we have 2 applications in these fields that involve water and it involves the manure. So it's quite a type of diffuse pollution in the area, and the groundwater system is very simple. We have the river, that's the upper part of the river, the ocean, the Mediterranean Sea, ocean. The Mediterranean Sea is on the right, and in winter we have, following the potentiometric map, we have a recharge from the river, and in summer, because of the groundwater extraction, we have a severe drawdown, and it's quite a type of flat surface. I will imagine that because there are tens or hundreds of wells pumping, then the flow field there will be a mess. So trying to follow the path of a contaminant into this aquifer, try to model it, we are trying to do it anyway, we are really optimistic, but from a common sense point of view, I mean, we will not be able to track this messy flow field and its influence in the transport that came from hundreds of wells, agricultural wells, pumping at the same time. Okay, this is the flow field, but indeed it's very simple. In the upstream, we have a recharge area that goes into the aquifer. It's like kind of a triangular aquifer, and in the lower part, it's the aquifer that recharges the river. Indeed, we have a very nice hydrogeological system, simple. This is a tomography that shows that the upper blue layer is the aquifer, which is very well defined, the one we have been sampling for this project. We are also monitoring the water table and continuous monitoring the water table. But going back to the flow field, the idea is that, okay, that's the chemistry, the real chemistry, represents the flow field. And it's, I mean, what is shown here, the triangles, the red one and the yellow ones, are those samples in the river, in the surface water. The white and purple dots, which are in the left part, are the sites of the alluvial formation. And the blue dots are the ones in the middle. What we see is that, for example, sulfate. Why sulfate? Because this surface water came from an evaporitic rock basin where there is a lot of sulfate coming into the water. It's not in the fluvial aquifer, therefore, if there is recharge from the river, this recharge might have a large sulfate content. So we see that the upstream dot, which is, the upstream triangle, which is the red one, is the one that has the largest sulfate concentration, and all the blue dots, more or less, can be understood as a mixing between this recharge from the upstream and the water that came from the side of the aquifer. So if we see that, we say, okay, it's interesting. We have a hydrological system with a river that fits the aquifer in its upper part. So, okay, if we look at going to the topic, if we look at the antibiotic content of, no, that's the one, of the samples, the groundwater samples, the surface water, we said the surface water has almost 1 order of magnitude concentration larger than that of the aquifer. So, considering what I've just said before, we might think, okay, it's the river. Why the surface water? Why the river has such a high concentration? Because of the effluent of treated wastewater that came from the villages we have upstream. That's our interpretation. That's a fact, indeed. But we can see from a hydrological point of view, okay, because we have this river-aquifer relationship, it's clear that it's the river, the one that brings the antibiotics into the aquifer. But if we think in that way, we are just misleading two options. One, that fertilization occurs all over the plain, and this fertilization using swine manure will bring some antibiotics also into the soil and into the subsurface, and we forgot about the geochemical behavior of all these pollutants. And these are the two things I would like to explain. First of all, of all the 53 components that were analyzed, only 6 of them were found at, let's say, representative concentrations, and they were usually found in the wells. Not all the wells had antibiotic concentrations, and not all the wells had the same proportions. That's the thing I will discuss later on. But we can say that probably the impact of antibiotics in this aquifer can be represented by these 6 compounds. When we map, because our hypothesis at the beginning is fertilization with swine manure will be the main source besides the river. So we should plot nitrate, which will come from the manure, and the antibiotic content. Here the antibiotic is represented by the antibiotic content is represented by the size of the circle. And the green color means low nitrate concentrations, and red color means high nitrate concentrations, expressed here as nitrate, not as nitrate as nitrogen, as you have usually seen in previous presentations. And we see that there is no relationship between nitrate, or there is no strong relationship between nitrate and the antibiotic content. Even though the source could be the same, why should we think that there is such a relationship? Both nitrate and antibiotics are reactive substances. They will change, they will modify their concentration once in the subsurface. So looking for this linear relationship, as have been said, have been seen in some papers, I think it's meaningless. If it exists, very well, but if it doesn't exist, it's inconclusive. But the most interesting finding at the moment of the project is when represented these 6, or in this case 8, more important antibiotics found in groundwater in those pie charts. And we see that it's quite heterogeneous, it's quite, how to say that in English, complicated distribution. We don't see any pattern. For example, if you see the points in the river, which will be more or less the points here, you have the brown color dominating, but here we have the yellow one. Here is the yellow one, and here is the mixing. So, I don't know if you hear me from below. So there is no spatial correlation. So what does it mean? It means that if we had a hydrological control of the system, that means the recharge from the river to the aquifer and from the aquifer back to the river, and more importantly, if the input concentration from all the funds was the same, and that's a very important point, then we should look some kind of pattern, hydrological pattern in the aquifer, but there is none. There is none. And if you look for spatial correlations, you do variograms. When you do the variogram of conductivity and nitrate, and I use nitrate, even though it's a reactive component, but we see that we have a range of correlation about 1,500 meters. But when we do that for the antibiotics, the most present antibiotics, which are ciprofloxacin and sulfamethoxazole, we see that the range of correlation is just 150 meters. Which, when you represent the variogram for the total amount of antibiotics, we see more or less the same range of correlation, which is more or less a pure nugget effect, meaning there is no such a spatial correlation. Why this? Well, we forgot to see at the geochemical part. It's well known, if you do some research, that all these compounds have a large coefficient distribution. That means that if we calculate the retardation factor that will affect its transport, or their transport, it will be very large. For a coefficient distribution of 5,000, the movement of these products with respect to that of the water will be very slow. What does it mean? It means that for each well we sampled, the area of influence to the well is the one that's just near the well. Otherwise, if the river recharges the aquifer and has some pollutants in it, they will just stay in the nearby the river. They will not move 1,000, 2,000, 5 kilometers away from the river. So the hydrological control is somehow affected by the high retardation coefficient of all these compounds in the groundwater transport. Here what I did is just to take the Ogata-Banks equation, that's the plot that's in the USGS paper, but I just added the retardation factor, which controls the velocity. And we see that for a given flow field and different retardation factors, the concentration that we will find in a well at a given distance, in this case 100 meters, may change a lot. Well, that's geology 101. But in our case, it's saying if our compounds have such a strong retardation factor, we should not expect larger concentrations of them at a given distance, at a large distance. And here, there is another interesting point. This equation has a boundary condition that says we have a constant initial concentration. And probably we think that this is not true. I mean, when we talk about antibiotics coming from fertilization, each farm has this manure pond, or pool, which the manure stays there for many months before it's applied to the field. What happens there? Which kind of antibiotics these farmers has given to the pigs? Will not be the same ones than the other one, or the other one. How is it applied? I mean, the initial concentration in amount and in quality will be different. So this explains the high spatial heterogeneous distribution that we have found in the pie chart of the representation. I have just 2 minutes, don't worry, Chris. Talking about the antibiotic-resistant genes, we look at them, the microbiologists look for these specific genes that are referent to specific antibiotics, and yes, they found them. They found them, and they found that there is also a difference on these antibiotic-resistant genes if we took the samples we took on the fall and those in the winter. We are waiting for the results of the spring, and the summer campaign, I think will be conducted next week, or the other one anyway. So, back to the conclusions. I mean, the point is, we have observed that the geochemical control and the IPUM concentrations will be more interesting to understand the antibiotics we find in the wells than the hydrological setup of the aquifer itself. We have to take care of the 3 topics: hydrology, which will be probably the less important one, geochemistry, which control the retardation of the solids in the aquifer, and then how is it applied, in which concentrations. That means talking to the farmers. That's the thing we haven't done, well, because it's just been working this for a year. But this poses several problems. The problem now we are discussing in Europe is how do we sample the aquifers in a representative way. So with a few wells, we get an idea of how the aquifer looks like. For nitrate, well, I will believe in an aquifer, an isonitrate map. I will not believe an iso-antibiotic map. Why? Because each well, at least in this area, is representative of itself. And that poses a serious problem on how to sample economically and representatively the antibiotic content and the pollution level of this aquifer. Okay, thanks.
Questions? Yeah, okay. So given the variability that you find, very nice talk by the way, Pep, very much enjoyed that. Given the variability that you find in your antibiotic concentrations, in your antibiotic resistant patterns, what do you suggest as an alternative to mapping concentrations as a way to provide information to decision makers? And maybe a first question to that is, how meaningful is it that we find these antibiotic resistant genes in groundwater?
To the managers, to the administration, there are 2 questions. What and why? And they will be interested in what is there. If you are interested in what is there, just sample those wells that are devoted or that will be used for public water supply. If the amount is very high, then you have to do something. If the answer is why, then you need a real aquifer study, hydrogeological, hydrochemical study to see what is the distribution in all the area. And you also need a lot of talking with the farmers to see which compounds are using. And also a direct measurement of what's in the manure. Knowing that sampling the manure and identifying which phase will be representative of its antibiotic content can be really, really difficult. There's a person in our team that is working on this. When she goes sampling, we all know in the institute, okay, because the smell of the manure just fills the whole building. The other question, what was the other question, Thomas?
How meaningful is it to have antibiotic-resistant genes and whatever concentrations you find it to public health or to your decision makers?
I haven't looked at this at all. I mean, microbiologists are still doing the laboratory stuff. and we haven't had time to discuss about the health concern of these antibiotic resistant genes in the aquifer. What is true is that, and they are really sure about that, is that the groundwater, I mean the bugs existing in groundwater show that they have been affected by these antibiotics coming from fertilization or coming from the river, mainly from fertilization, I will say. But the health concern about this, I cannot tell you.
There's time for one more quick question. There was one.
We find a lot of antimicrobial resistance genes in systems that never receive manure too, right? There's many pathways by which these things go into systems and they get transferred and they move around, bugs try them, kick them out. They're everywhere. And some of these things could be misnomers as well. And I think in our study that I presented earlier, we were kind of lucky in finding one AMR gene, just one, that we didn't see in the soil or in the water previous to application. But there are others that we see continuously in the system. And so that's important. And also as far as, and you're right, and like the manures, you can take a sample here and here and here and have a whole different profile, a community, different populations of genes that are in there. And so I think you're absolutely right in that probably the best place to go within the context of decision-making endpoints are, because these do have public health risks, obviously. I mean, AMR is a big deal right now. And, you can get these things in systems and, you get a lot of resistance. But knowing what's in the manure itself, I think, is important. And I think moving away from use of antibiotics, of course, or using vaccines and so on are ultimately what we want to be going at.
Thank you. I mean, regarding the relationship between antibiotics and antibiotic-resistant genes, the microbiologists, what they do is you may know, really well known, they look at the specific gene that is a proxy for the pollution of such antibiotic. So we can say that there is a direct correlation between the occurrence of this antibiotic and the resistance shown by the resistome. And then, I don't know what else you said, excuse me, I lost it. No, and regarding the heterogeneity you mentioned around, that's a discussion, I don't have any conclusion about this, about other people who works on artificial recharge, using treated wastewater without a tertiary treatment. And they also observe this kind of a heterogeneity, and that the emerging contaminants, not only antibiotics, but the other type of emerging contaminants, also appear with a high degree of heterogeneity. And one of the questions under discussion is if the bacteria, the bugs, they move around the aquifer, or if they are more or less in the same spot, and they change according to the quality of the water they get. I don't know if you can, it's me who puts the answer probably from now at the moment, but that's the discussion we have now in Barcelona with people who does this artificial recharge. Okay, thank you.
Thank you.